All right. Hello, everyone. Hope you had a great lunch. We're back for block 2, this time stories of data reuse. And our keynote speaker this time is going to be Komenda. Martin leads the research team at the Institute of Biostatistics and Analysis of Masaryk University and serves as the national coordinator for health data at the Czech Institute of Health Information and Statistics. He will present health data stories. Please welcome Komenda.
Thank you very much for a nice introduction. So welcome back from the lunch. And thank you very much to organizers inviting me. Well, what's the point of me being here today? I'll be quite happy to share with you our journey, our experience, our story based on health data, health information, the communication with lay public as well as professionals. And must say, honestly speaking, this is not quite easy for us. But we're still struggling, but hopefully I'm gonna share with you our experience as well as pitfalls. I've been already introduced, so shortly, my academic home, Institute of Biostatistics Analysis, under the umbrella of Masaryk University. I'm quite proud to stay here. And I'm also leader of Communication Data Center of Institute of Health Information and Statistics, which is under the umbrella of Ministry of Health of the Czech Republic, so I'm quite close to the top management of Ministry of Health. Well, let me start with going back for a moment. You probably know this very well-known man as a pioneer of computer science, because it's Tim Tim Berners-Lee, who is founder or co-founder of World Wide Web. And 10 months ago, I watched a very interesting interview with Tim BL and his close colleague. And here is the message, which I found quite interesting and motivating for me. So I'm gonna put it into the slide. And this is just the very beginning of my speech, because in the conclusion, I'll be back with our additional value, and hopefully in the terms of health data, health communication, I try to tell you more. Okay. Everything what I try to say today is touchable, it's online, so if you wanna double check or just to have a look, please visit nzip.cz. Sorry for foreign guests, very nice guests. This is only in Czech language, but hopefully I try to explain the main mission. And as I said, our activities are not just projects or for funding. This is just for, hopefully, benefit for the Czech Republic and for the communication of health information and health data. Well, I think each habitant, not only the Czech Republic, deserve the platform as a central gateway, as a, let's say, small Google, which will be trustworthy, definitely high quality, with no manipulation, strongly non-political. And I think we built something like that. This is the homepage of National Health Information Portal as a central gateway, with the primary purpose from 2020, when it was being maintained firstly with the strong support of Ministry of Health. And from this year, we try to provide just one central guaranteed platform, firstly oriented for lay public, or for general public, with the communication of clear information regarding our health system, how it works together with, let's say, life situations, preventions, etc., etc. But the things were to be changed. So right now, we are speaking also about the communication based on data or health data, and the abbreviation NHIS Open means National Health Information System, as a central backbone of storage of all health data in the Czech Republic. And open like openness, like open-minded approach, like support of sharing data for all of us. Well, to be honest, one of big accelerator was the period of COVID-19, because the increased interest of not only professionals but also lay public in data, in health data, in trends, how it works on, let's say, municipality levels. And definitely for us, it was like amazing push because the system wasn't ready for this kind of crisis management. So we built definitely new information system and try to share new data reporting services, which is definitely, or it must be based on open data sets. Well, what we did, you can visit if you want from National Health Information Portal's Central Gateway, data reporting. This is definitely not about big data. You see the amount, the total numbers of open data sets, as well as data summaries and visualizations. Hopefully it is about the quality, about the guarantee, about the system what we decided to release, to share different components of National Health Information System. You can double click and you can see what is accessible, and I'm going to comment right now. Just for an illustration, I think you are not probably medical professionals or experts. National Health Information System is a really big agenda, consists of 60, 61, 62 agendas. You can imagine, for example, the biggest, let's say, registry, National Registry of Reimbursed Health Services, when you can find all bills, all counts regarding covered health insurance here in the Czech Republic. Administrative data, but quite fruitful regarding the reporting, the predictions, etc., etc. Together with evidence of health professionals, definitely, together with health service providers and many, many more. You probably know National Cancer Registry or National Registry of Cardiovascular System, etc., etc. Well, because we are a government institution, we need to have also some legacy background or some methodological background. And I'm proud to say that from December 2023 until May 2025, we went through a really in-depth review process when all the stakeholders commented our methodological background. And right now everything is published. Unfortunately only in Czech, but I hope it's quite complete. So right now we covered all these details, all these access regimes, how to release, how to share health data for individual stakeholders, individual entities who are in a process of data creation, data storage, and data reporting again. Together also with being, let's say, in compliance with GDPR on this legal background, which is quite necessary. Well, when we started with the communication of individual target groups, we created this schema. I like it because here you can see 3 levels of, let's say, understanding of needs or requirements. And right now I think we are speaking here on this floor with data explorers or data experts, but we try to speak also with just a representative of lay public, where somebody is used to consuming just on infographics, just one number together with correct interpretation, and it's not easy to create with the guarantee of, for example, experts from Czech Medical Sciences Society, which covers all these experts cross medical disciplines, but we try to do our best. You see the portfolio, different formats, different, let's say, levels of detail, but everything is under production in some kind of state, and I'm gonna share with you some stories. Well, regarding health data processing, this is not only about deep level of expertise in IT or data science, it's also about domain expertise, as our colleagues mentioned before a couple of minutes. So we prepare guided tour, let's say, where you got all the information which is necessary to adopt when you enter into the system of health data in the Czech Republic. You see again different formats, different style of information, like a moderated interview, like question and answer, together with official documents, methodological background, and some other guides how to go in this complicated process. Here, this is the homepage of our health data catalog. You can imagine, and you probably know, you are familiar with open data catalog as one of the, let's say, mandatory tools across the resource under the governmental level. And we did something, let's say, more extended, because local open data catalog is just one part of a whole data catalog. And you can see on the right-hand side where you can see the formats, like open data sets, which are strictly defined by law, by some characteristics, together with the connection, the relations, the harvesting with a national open data catalog. But we did more, because we prepare also another formats which are communicating with our target groups, and everything is indexing here. Like analytical studies, you can imagine just PDF reports with many graphs, tables, and interpretations. You can see also data summaries, which represent another aggregated overview of data in form of Excel file with a lot of sheets, which are divided into the municipalities or age groups, etc., etc. But what I'd like to highlight that these, let's say, raw formats of outputs like open data sets and data summaries are always extended by metadata description. And here we can try to do our best to communicate also information limits, also the information about the source, about the sources, what are the additional value, what was the primary motivation to create this particular data set or data summary, and we hope that it helped with the final orientation with the particular output. And last but not least, also interactive visualization infographics, which help us to deliver the key messages or take-home messages to public. Okay, let me shortly speak about 3 stories. First one is, just imagine you are just a normal habitant in Czech Republic, and you are curious because you plan to have a surgery, and you are curious about how many patients already passed the same procedure as you plan to have. Here you can also visit the catalog, and using just easy procedures or something like that in the Czech language, you can find and you can consume, for example, this interactive visualization. I like it very much because this visualization hides a lot of various overviews, because you can click on the filters and you can modify, for example, the municipality or age groups or sex or whatever else, and then you can specify your particular overview of available data. You can see also something a little bit different, but based on the same style. We try to adopt open data first, so firstly release open data sets and then build the first floor in a form of interactive visualization or infographics, for example. Second story, a little bit more complicated, let's say. Just imagine you are a journalist. By the way, very influenced person who can really share with the general public the results and influence the meaning in a society. And here, just pretend you are curious about the trends of the hospitalization in Czech Republic. And yeah, this is just the sample of hospital or medical procedures in a form of data summary. And again, different point of view regarding the total numbers, regarding the habitants in relative and absolute point of view, together with detailed documentation where you can read more about particular attributes, which help not only to the particular consumer as a user, but it also helps AI tools to understand the connection between individual data sets. And finally, story number 3, I hope the most interesting for you because you are experts. Let's imagine you are the representative of pharma business sector, like pharmaceutical scientist, and you do have the question like it's shown here, like the morbidity, prevalence, incidence, mortality, etc. But in this particular case, the result is not available in our catalog, so you need to use something different. And I'm proud to say here or present our system of synthetic data in a health domain, because what we did, and it was quite painful way, but we did it, hopefully successfully, we present here, and using QR code you can see more details about the methodological background together with the explanation how it works in practice. This system is like the solution for secondary usage of health data. I hope also answer for health, European Health Data Space Commission rules and what will come very soon, and how it works in practice. Let's start from the top. What we did, we put just 4 main components of National Health Information System, and then artificially generate the data which fully respect the structure, I mean data schema or data model if you want, then also relations together with entity and the attributes, and this is the step 0, let's say. Then we open the request system where you can formally submit this request. Then, as I mentioned, it's quite formal, so you definitely succeed. And then you are a member, let's say, or user of the system of synthetic data. Then you can access to the database, again, with fully artificially generated data, so you cannot achieve any real results which can influence whatever. This is just for you as a computer or data scientist to prepare and optimize your SQL script with some kind of additional value which probably solves some kind of real problem, like research questions or whatever. Then share with us the final script and submit your final proposal. On our side, right-hand side, then we need to go back to primary data, then run the script, and then we need to get the final double check or triple check regarding GDPR and this kind of legacy policy background to be sure that everything is correct. If so, then you got the result, and you got 6 months for presenting your, let's say, primary results in a form of paper, in a form of your business plan, whatever. And after 6 months, this output, probably in a form of data set, data summary, is published in a health data catalog, because we try to support in this way open community, open-minded approach. And then etc., etc., etc. From May 2025 until this day, we got, let's say, 125 users of system of synthetic data, and we have already managed like 75 requests or submissions. And today, it was the first day after the 6 months when we published really very first results in the health data catalog. So when you can visit the catalog right now, you see in the news the first 3 outputs generated from the system of synthetic data. I must say that the reports are on patient level mainly, and traditionally it helps to answer some research question. Well, just for your illustration, what are the sizes and proportions between the primary National Health Information System data and our system of synthetic health data? You see, it's just a really micro visit of all these databases. A schema, but definitely it works in practice, and on the left-hand side you see also the components of National Health Information System, for example, also death certificate information system, because you also need to know this information when you, for example, track patient pathway through very beginning till the end. Yeah. We also establish the communication channel for researchers, because if you are so open-minded and try to share the computational script, like SQL, typically hundreds or thousands of rows. You can do it, and for this purpose we established National Health Information System Open Journal with editorial board, and you can also open this know-how with the community. Must say, like, only 20% are the users who want to share this know-how with the rest of the community, but I'm happy that it's 20%. Why not? And yeah, I think next week will be the very first moment when these scripts will be also published as a part of National Health Data Catalog. We are also researchers or academics, so if you are interested in National Health Information System and the journey from very beginning through the optimization phase until now, everything is reported in this paper in JMIR Medical Informatics. I think the system deserves something like that, so have a look if you are interested in. And there are two papers under the review in this moment. First one is more policy making oriented, when we try to deliver some mission for another European countries regarding the system of synthetic data and how it could be adopted by some another policy-making stakeholders. The second one is also under the review and try to describe also technical background, the solution itself, together with some methodological backgrounds, together with legacy issues, etc., etc. And finally, what we did together also with colleagues which are sitting right now, thank you very much again, are some case studies based not only on medical and healthcare education, but also on health information and statistics. And you can try, it's also online, you can try to have a look, and then you find some case studies where a selected methodology was used to explore real data, open data sets which are published for general public. This is also the components of our regular classes on Masaryk University, when students can have some playground for data processing mechanism. Finally, let's go back for a very beginning. And I presented with you the sentence or take-home message from TBL. So let me shortly describe the modification. I think we can get value of National Health Information System as a small piece of human interest. And finally, health data is held in place in National Health Data Catalog, which is accessible for us, definitely, together with persistent links, together with some long-term usage, and this is the way of our understanding. And finally, thank you very much for coming, for listening, because you are also maybe not health, but you are also open community members of open science, and this is the way which I really like. So thank you very much.
Thank you so much, Martin, for the interesting presentation. Very important work that you do. All right, so we're on to the flash talks now, and our first flash talk is going to be from Jaroslav Oľha, Faculty of Medicine, Masaryk University, and he's a bioinformatician with a background in computer science and biochemistry, specializing in translating complex data into actionable biological insights. He's going to be presenting on using and reusing single-cell RNA sequencing data. Please welcome Jaroslav.
Like this? You said, and don't swallow.
Don't swallow.
Okay. Hi, thank you for the invitation. I'm happy to be here. Sorry, the lightning talk is like my kryptonite because I tend to ramble on, but I will try to keep it focused for you. I'm from the Department of Histology and Embryology at Faculty of Medicine at Masaryk University, and I'm here to talk about some of our data, and the largest and most interesting data we have is the single-cell RNA sequencing data, which some of you may have heard of. It's like one of the most data-intensive and most widely used data methods in biology right now. I know we are mostly here to talk about data use and reuse and storage, but in my mind it doesn't make sense to talk about data in isolation, so I will spend some of my precious time trying to explain what single-cell RNA sequencing is, and I will start. Oh, this works great. With this helpful little box down here, this you probably know, it's a very famous image, arguably the most important process in life. DNA famously stores your genetic information. It's what makes you you and what makes you different from blade of grass, and it does so by encoding proteins, which are tiny biological machines that are responsible for just about every process in your body. And what you also probably know is that there is an intermediary molecule called RNA, or mRNA in this case, because the DNA likes to be packed tightly and safely in the core. So whenever a protein is needed, a gene is activated, this little molecule copies the genetic information and carries it wherever the protein needs to be made. Sorry. Knowing that, I will ask you a question that you can answer in your head. What is the difference between DNA sequencing and RNA sequencing in terms of the data we get? This is a lightning talk, so you get 2 seconds. 1, 2, well done. I'm sure you got it. The difference is that DNA tells us what kind of genes could be activated, what kind of proteins could be made. It's the source code, it's the blueprints. The RNA actually tells us which genes are being made, what kind of genes are activated, what kind of proteins are being made, and what is actually being carried to the protein-making machines to make the proteins. So if you give me a bunch of cells and I sequence their DNA, I mean, I can tell whether they're your cells or someone else's, but that's about it. But if I sequence their RNA, I can actually tell which genes are activated, what kind of cellular programs are enabled. So it gives me kind of a snapshot of what the cell is actually doing. So you give me a bunch of cells, I sequence their RNA, I can tell you, oh, these are brain cells, some of them are neurons, some of them are astrocytes, they seem to be fighting some kind of infections. So RNA sequencing is actually the name of the game in most biology labs where you study diseases or some kind of dynamic processes. You always sequence the RNA, which tells you the dynamic state of the molecule. That's for the RNA sequencing. Now, single cell doesn't mean we sequence just one cell. We actually sequence a bunch of cells. If there's one thing you remember from this presentation, is that single cell sequencing doesn't mean that we are sequencing one cell. What it means is we get a single cell resolution. So we know for each cell in the initial mix what its genetic profile was. Because to do RNA sequencing, you actually need to kill the cell and dissolve it to release all the RNA, and only then you can sequence the soup. And you need to sequence a bunch of cells to get a strong enough signal, so it gets all kind of mixed up and smooshed up together in one big soup. But single cell sequencing, through genius bioengineering, allows us to actually, before we dissolve the cell, we label all of the RNA in that cell with a barcode. So even though we dissolve all the cells, create this huge goulash, we then, in retrospect, from the data, we can piece together which pieces of the RNA came from which cell, and we can actually get this kind of a matrix that for each cell in the data set, which there are tens of thousands of them, tells us the expression of each gene in each cell, which, again, tens of thousands of rows. And what we then do with the data, obviously, is we somehow reduce the dimensionality. Each dot on these plots is one cell, and we group them together based on their similarities, based on their expressions, and this allows us to, this is just a quick example of one of our more recent data sets. This is healthy brain tissue. One of our specializations is growing tiny brains in dishes. So we grow brain cancer in a dish, we grow a brain in a dish separately, we see what kind of expression they have, what they do, and we also, in parallel, we grow them together, so we squish We kind of infect the brain with brain cancer, and we see what happens. They're tiny, they don't feel anything, and this immediately visually tells us what kind of new cell populations appear as a result of the tumor invasion. So we can immediately zoom in on the cells that are clearly the tumor reacting to the healthy tissue and the healthy tissue reacting to the tumor. If we didn't have single cell resolution, this would all get lost in the noise, because most of these cells are just brain being brain and tumor being tumor, so we would lose these tiny pieces of signal, but now we can immediately zoom onto the areas that are responsible for the entire mechanism of the cancer infection. So I hope you can see how this is incredibly useful and information dense and deep inexhaustible data. Like, we have been looking at this data set for almost a year, and we are not done. It's just a huge rabbit hole. Now, for my final slide, the challenges. This will be really quick. Our challenges with this data, again, the data is incredibly information dense, endlessly reusable, if you can interpret it, and that's our biggest problem. So our problem is not incompatible formats. Those are fairly standardized, just basic sequencing formats that have been here since the early days of DNA sequencing. And it's not unavailability of data. Like, scientists don't mind sharing their data sets. Usually they publish their data with the paper, but the problem is putting it together. There's a huge amount of technical noise in the data introduced at every step of the process. There is noise from the initial biology, how the samples were grown, it's from how they were collected, how they were sequenced, and how they were processed, and that's the problem. Like, here I should have introduced these are 3 different data sets of cells that should be the same general kind of cell, and they just don't overlap at all. And you can use various integration methods to try to fit them over each other, but it will not always work. And the reasons, like the problems we need to tackle to get to a point where we can really reuse the data and squish different data sets together, is one, the tools, the mathematical methods for integration. That's a huge obstacle, but it's also annotation, because we're not sure if these are the same kinds of cells. Like, the paper says so, but there's no kind of annotation, there's not enough metadata. We don't know if what they call neurons or mature neurons and what we call mature neurons, if that's the same thing. We don't know how old they are. Like, there's a lot of data missing, and we need more information to be sure how to actually integrate the data. And also its dynamic states, we don't know. It's really a bit of a mess. But the problems, like, the thing that makes me kind of hopeful is that the problems, to me, seem like problems that can be solved in a centralized way. Like, I think those are the problems for rooms like this one. If the problem was that people don't want to share their data, or everyone uses different data formats, then that's, to me, would be a difficult challenge, because you would need to persuade thousands and thousands of scientists to do things differently. But these are all, I think, challenges for repositories and infrastructures to actually take all of the quantities of data that are out there and help scientists put them together. Thanks, sorry.
Thank you so much, Jaroslav. I was just gonna say thank you all speakers for keeping the time. And it was great, I liked it. All right, let's move on quickly to Monika Čechová, who is from the Faculty of Informatics at Masaryk University. Monika is an assistant professor at the Faculty of Informatics, researching complex parts of human genomes and applying machine learning to genomics. Please welcome her presentation, Experience with Data Sharing and Reuse in the Field of Computer Science. Give it up for Monika.
So I will start by disappointing you because I'm actually also a biologist, not only a computer scientist. So today I want to talk to you about my experience working as part of the consortium. It's a human pangenome reference consortium. And what I will be presenting, hopefully, is not what I would claim to be like the best practices, but I would call them like practices like good enough that have been successfully applied to this project. So genetic information is extremely important because when we study DNA, it really helps us understand human diseases, including cancer. So lots of these diseases are encoded in our DNA, or the variants in our DNA increase the probability of us having those diseases. At the same time, we're lucky because DNA is really just 4 letters, A, C, G, T, and that essentially means that like our genome is just a text file. And it's a pretty small text file. You can store all of your chromosomes on like not even a CD, like it's just 6 gigs of data, and that's all you need. Okay, so let's speed it up. So we all know what DNA is, right? So we need lots of DNA. And before we understand the disease and the DNA that causes disease, we need to understand the healthy variation. We need to understand how DNA looks like from humans all around the world. And this is the goal of the Human Pangenome Reference Consortium. Disclaimer, I'm not the leader, I'm a member of this consortium, and so this is experience from like a member's perspective. And the topic of this session is reuse. And in order to do this, we will start by reuse, but not the reuse of data, but the reuse of material. And so almost 20 years ago, there was a fantastic initiative called 1000 Human Genome Project, which did sequence human individuals using the old technology available at the time. But that material is freely available and serves as a rich source of information how DNA looks like, what human variation to expect, what mutations do we all share, what mutations are common, what mutations are disease-causing. And so part of the approach of the consortium was to use this genetic material, use this biological material, but this time read it with the latest cutting-edge, state-of-the-art algorithms, and basically provide complete human genomes that then thousands and thousands of researchers can use all across the world. So basically everyone can use this rich data set. Human Pangenome Reference Consortium was started in large part thanks to Karen Miga, who I was lucky enough to work with as a postdoctoral scholar at the University of California, Santa Cruz. Okay, so lots of different data types that if we put them all together, we will get complete genomes of many individuals. So how do we actually do it in practice? So all the data is stored in AWS on the Amazon bucket, and all the wrangled data is stored in a table. So if you're our collaborator, we're gonna give you a table that is versioned on the GitHub, and in this table you will have links to all the data sets for the download. You can also see the GeneBank accession, which means that the final products, the final DNA is also stored in a shared repository that is used by all the researchers. And we also have MD5 sums and all the information you can think of. This is how the table looks like. You can see that there's a lot more metadata about the sex of the participants, the population they are coming from. This is really data from all around the world. And you can also browse the data manually on the AWS bucket, and you can even like download it by right-clicking and download if you should feel inclined to do so. This is the HPRC Data Explorer, where you can filter different data types and download the data yourself, or just explore what's out there available to you. And the big motivation why all of this data is available is because these participants really were so fantastic that they gave us a broad access so that anyone can reuse this data, anyone can do any research project on it. And it's really thanks to this that we have a low barrier of entry for also international partners, so anyone can join and analyze this data. And this is no small feat because at times this is really like petabytes of data, and that's also why it's important that we also offer centralized analysis and pre-processing. And so, for example, instead of dealing with these like hundreds of terabytes of data, if you're studying genes, you can just download genes. If you're studying mutations, you can study mutations, and you can really benefit from somebody else doing all of this pre-processing for you. Some of the open science strategies. All the raw data is completely open access for download. All genome assemblies are versioned and uploaded to the GeneBank central repository. All the code is open. All the workflows are in another repository, DocStore, and all publications are pre-printed and on bioRxiv. The lessons learned, the data management can be a full-time job. Heterogeneous data sources add lots of overhead, so if you have lots of international partners, that can be challenging. And again, the open consent really helps a lot, and to coordinate, it's also very useful to come up with some weekly checkups and communication channels. And with that, thank you for your attention.
Thank you, Monika. All right, up next, we have Marek Cebecauer. He is a biochemist engaged in interdisciplinary research with a focus on imaging immune cells and the data management challenges that come with it. He comes from the Jaroslav Heyrovsky Institute of Physical Chemistry, and he's going to have a presentation on I Found It, Automation Accelerated by Data Sharing. Please welcome Marek Cebecauer.
Hello everyone, and I'm here to share with you the solution. We sorted all problems. I'm not blonde enough, sorry. I'm going to share with you our new experience with a solution we hope to be for many of you. The fantastic day started with the fact that I have access to the data. And this would, of course, not be surprising, but I can do something with this data and use analytical tools to make it black, to produce something useful. Or I was introduced as a biochemist and immunologist, but in data science, I'm more involved in helping colleagues from material science. And they are using computational analysis to come up with a design of new materials, to produce something innovative and black, and some new products. Again, this would not be surprising. This is something many of you are doing, but how this happened? Again, black. My finger is too big. With colleagues from Liberec, we are developing a system DDR, and in this system you can log in using your standard EduID login. And in the system you can organize, you can book instruments, you can do your measurements, you can access your data sets, you can manage your instruments, and so on and so on. Importantly, you have a DDR browser, you can access your data. So what happened is that one of the colleagues measured the data and made them available. This is Jakub, and I am Anna, and I am accessing the same data in one click. And this is very important. This is now really happening, that I don't have to go to Jakub. I can just open DDR, and all the data he measured yesterday and analyzed this morning. In the afternoon, I can work on it and really design new things. Even the system allows that you convert the data, you pre-process the data, and I can still, as you see, the original, some spectrum, infrared spectrum can be processed into the formats which are better for me, and then I, as you see, I can see it. So what the system is, the system I was presenting a year ago, is helping. It's very simple, that's very important. It's very relatively easy to implement, and there will even be a SESNet instance, so-called light instance, because for automated harvesting of data from instruments, there is need to do something on your side, but we want to also share one instance which will be available for everyone on this in Czech Republic for researcher, and they can share the data using this system. The importance is that they don't share only the data but also the metadata. That's important in this case. So the DDR system brings research from its standard way. When it's relatively slow, it's performed in silos. Then the DDR team was a little bit thinking, then a lot of coding, then testing and implementing, and we have now a solution for the today's science, when if you have someone to implement it in your institution, you can have automated data harvesting. There is no paperwork associated. That's very important. My DNA colleagues know what does it mean, the legislative issues. And there is instant online data access to the data, and there is reliability in the system. Thank you very much for your attention.
Thank you for your presentation, Marek. Next up, we have Adrián Rošinec, our last lightning talk. He is a researcher and IT specialist at CEITEC, focusing on research data management, fair data implementation, and developing a national repository for molecular dynamics simulations. He's going to have a presentation on data use cases at CEITEC. Yes?
Yes, thank you, and hello everybody. I'm Adrián, and I would like to show you some data use cases that we have as a core facility at the CEITEC at the Masaryk University. So first of all, what is the CEITEC? The CEITEC is a consortium of several universities in Brno and Academy of Science. And we have at least 2 buildings. Within we have several research groups focusing on different topics, and also shared laboratories, so-called core facilities, that are providing the instruments and all this kind of equipment to do some measurements, experiments, and to support research with the data. Which means if we have such variety of the institutes in the consortium and data, like creating machines, we need to come with some solution that will aggregate this, the access to the data, and also somehow support the sharing and reuse of the data that are being created in here. So this is basically an overview of how many core facilities we have. These core facilities have several devices, experimental devices that are producing the data. So this is just to make some overview. I'm from the biodata core facility, which is developing the solutions at the CEITEC for all other core facilities to provide these data management systems. I'm here on behalf of Radka Sovodova, which had the scientific advisory at the university, because she obtained it today, the professorship. So let's congratulate her. And this is basically what we are doing with her. It is the data management system based on the one data, and our idea is basically that the user or the researcher of ours shouldn't at any case come into some technical aspects of the infrastructure that is provided by the national infrastructure, or if the national data infrastructure will be provided, then with the technical aspects of this. And I mean by like working and playing with the data transfer, planning it manually, planning some data transfer from the instruments to the computing resources, etc. So we are providing something, or we are trying to provide something like the system that unites this workflow. So basically, at the beginning, we have some instrument, we have some device that is providing the data with the user that is doing some metadata annotations within some console PC or somewhere in the electronic laboratory notebook. And then if the data set will get ingested into the data management system, it is or it should be available all around the portfolio of e-infrastructure or the national e-infrastructure services, such as, for example, the Metacentrum, the Kubernetes, the Jupyter notebooks we are providing, the Galaxy service, and also it should be easily like uploaded or transferable into the data repositories which are being now built for thematic communities. So this is our basically idea, and we are working with within the CEITEC with the colleagues from the infrastructure on this, and we have several use cases that are already using this system at the CEITEC, and they are getting the benefits out of it. So this is the MAFIL laboratory. We had a colleague, Dr. Fosek, from this laboratory. They have several instruments that are concurrently measuring the patient data, and this is some, sorry for the translation, but this is some life cycle of the projects itself, because when some group of scientists will come for to the Czech bioimaging infrastructure, saying what kind of or what type of data they want for their research, they will be asked or discussed with the technical parameters and the preparation of the process. They will be asked what kind of data they basically want, and the core facility will start finding and booking volunteers and placing them appointments with the MRI basically, and provide the research group with the measured data. This includes basically some kind of the processing of data, as Honza mentioned, that they are doing also some analytics and post-processing for the data, but also proper sharing of this data. If they want, the users or the scientists can get only some DOIs to the repositories where they can find post-process data, and they don't have to like handle this by themselves. So this is just how some project details looks like. It is full of some metadata, ethical committee data, and this kind of stuff. And then we have some life cycle of data which are coming from the MRIs, from the user surveys or participant surveys, also some measuring protocols. So this is all one big data set consisting of several visits from the several patients in the MAFIL laboratory. Then we have some data catalog basically service, or the information system, where everything is basically registered and available to the users, where they can download the data, annotate the data, and yeah. So this is way how to support basically this laboratory. Then we have more generic solution for the data. This is for the cellular imaging laboratory, and we have several optical microscopes. We have some processing servers for the size images which are coming from the microscopes, And then we have this one data system that provides these images to the users, but also provides these images to some visualization and analytical platforms which are deployed then in the infrastructure. User is only telling one data basically which kind of data set he would like to analyze, and the one data will manage the transfers of the data sets to the hot storage of the computing platform basically. And last thing, sorry for being so long, but last thing is basically providing, these are the things that we are doing basically for the CEITEC research groups. And the last thing is providing the molecular dynamics simulations data repository. When we have some nice federated model where there is one European catalog for this data, and we are one of the nodes because we have very large data. We are proposing something like the 5 petabytes of data. And I wanted only to show you how we are going to implement it. Yeah, and I also wanted to say that, sorry, just last thing. Okay, let's this for the discussion. But yeah, sorry.
It's okay. Give it up for Adrián. Thank you very much. All right, we are now done, and we are ready for our Q&A chair, which is coming up now. Please welcome Jaroslav Juráček, the deputy head of the Czech EOSC Secretariat. He leverages his expertise in biochemistry and oncology to help develop research data management in the Czech scientific environment. Within the Czech EOSC initiative, he focuses specifically on the challenges of sensitive data and establishing secure access frameworks. Please welcome Jaroslav Juráček.
Thank you so much. Thank you for a nice introduction. And first of all, Monika, Marek, I'm so sorry for the technical problems, and I will try to give you extra minutes during the discussion. On the other hand, I might take a few minutes from Jaroslav and Adrián. Anyway, before we start to share some more data stories, I would like to take you for a short trip, specifically to the 16th century, because there we have two famous scientists, and they perfectly represent the theme of this section, and they are Tycho Brahe and Johannes Kepler. While Brahe was a brilliant observer, and for decades he tirelessly generated data which were unique, were most accurate in his time, and were basically the best you could get. But he lacked the, let's say, tools and maybe even knowledge to uncover some deeper loss within this data. So basically, what he generated, or maybe what he created, was a perfectly curated and documented data set. And then enter Johannes Kepler, and he was a genius mathematician. And he gained access to this data set, and he spent years to analyze it. And then it was based on Brahe's data set, not his own data, that he formulated the 3 laws of planetary motion, which actually changed our understanding of the universe forever. So for me, this is a perfect example, data transformation into knowledge. And why am I talking about this today? Because I think that such Brahe's data sets can wait in every research laboratory. And the challenge is actually to ensure that they won't sit silently, but they are prepared to be found and understood and reused by research communities. And therefore, my first question will be very simple. Do you use external data? But I will make it more specific. If yes, please tell us how often, what is the data source, and maybe you can also specify whether it's data curated, non-curated. And I would start with Adrián, and maybe and with Martin, because I have a feeling that I might be I have to be more specific with you.
Yeah, thank you. I'm more in the role of like handling the data if they have been already produced or generated when talking about the molecular dynamics, and doing this, handling those data for other scientists. So I'm not in the particularly, like I don't have that much experience with like taking the data from some data repository and using them. I'm more into like putting data into the repository. But if we are talking, for example, within our research group for the structural biology, we are using the PDB database, protein data bank. We are using the structures for like creating the molecular dynamic simulations, or for doing some analysis on the protein structures. So yeah, we are using the data.
Yeah, I'm actually explaining every research in our institute when they answer. We have our own DMP form, and first question is what data will you reuse? And common response is no. And I'm usually, my response is no. So I can't imagine to do research without using some data. As a research PI in immunology and biochemistry, I used omics data, structural biology data. In material sciences, it's more crystallography from crystallographic database and properties of some elements or materials which are in public. All what I mentioned are public databases. Unfortunately, in my field, in the imaging of immune cells, we share zero data. My lab now shared some data openly before publishing. It's probably the only data set which exists on this planet in this particular field. So there is a lot to do.
Thank you.
We use data all the time, and I feel like genomicists and bioinformaticians have done like wonderful job, not only like filling these databases, but also coming up with new algorithms to query them quickly, to efficiently store them and save space and all of that. And the biggest challenge really is metadata, that like you find this like amazing data set, and even if you read the associated paper, you would still love to talk to the person that generated the data and ask that like tiny little detail that might be critical for your experiment.
Thank you, Monika.
Thanks. Yeah, we also reuse data, but we mostly do it in this prehistoric way where you read the paper and you have to find out what the data is from the paper and hope that it's thoroughly described. But yeah, we mostly use it to validate our own data. Our own data is always the main focus, but it's a mess. There's a lot of ways to interpret the data, so the data set usually isn't strong enough to make any claims on its own. So then you find papers from groups who do similar work, download their data set, but it's not like integration of a bunch of data sets that we find in the database. You always find the paper, find the data, try to find out from the paper what the data might be, and use it to support your own claims.
Maybe I will ask one more question. Can you imagine to do your research with single cell solely based on external data?
I can imagine it, but it would be super hard. But there are definitely bioinformatics groups that don't have a wet lab component. Like we are mostly a biology team that is just dipping its toes in bioinformatics and data management. But I know there are just purely bioinformatics groups that work only with external data. But that sounds like really difficult. I wouldn't want to do it that way.
Okay. And Martin, the core question stays the same. Will you use the external data in NIH-IP? But by external data, I mean research-held data. Is it possible to integrate research data as a data source for you?
So in this particular moment, honestly speaking, this is not our experience or our way. But generally speaking, about external data, we got strong and long-term connection with Czech Statistical Office because of demographical data and trends, definitely. And one very challenging agenda is right now to connect with, let's say, social sector. For example, for mapping palliative care in the Czech Republic, and you need definitely health data from, let's say, administrative part of you, together with data from social sector. Generally, it's not easy because sometimes it's a little bit like a digging somewhere and try to find something, but the very first results have been already, let's say, published or released, and you probably know you've seen some shows of our director, Professor Dušek, because this is quite challenging also regarding the budget and investment on the floor of the Czech Republic and the budget itself.
Excellent, thank you. So what I can see, using external data is a good practice, and basically all of you have experience with that. And for me, good question is why to do it. So I would like to ask what might be good motivation for data reuse. Whoever wants to start.
Let me start with the micro. I think the best motivation are real use cases. So let's have research question or some kind of motivation behind what trying to answer or try to explore, and then it's for us it's definitely like the definition of existing of each individual registry as a part of National Health Information System, because you need to prove the sense of the registry, and the first step should be definitely sharing some, let's say, basics from the registry, and the second step should be like the connection with the rest of the components and try to predict something more, let's say, sophisticated.
Thank you.
For us, kind of obvious, it's free data. That's often just as good as yours. For us, data is usually used as kind of a source of information and source of hypothesis for wet lab experiments. So it informs the practical experiments in the lab under the microscope. And you can do that just as well with someone else's data. Like the reason we generate our own data is because no one is performing the experiments we need, but if some other lab happens to be exploring the same hypothesis and the same biological systems, their data would be just as good as ours. So for us, someone else's data can also generate hypotheses for the biologists.
To me, it's like equalizing aspect, like anyone can do science, anyone can make a discovery. High school students can like download DNA and like learn anything. So and it's cheap.
Okay, so I agree with colleagues, of course, but as I said, in our field it's not common, so we maybe even like to share the data to train the community, to change the habits in the community. And also one has to say that we are fully public, we have a fully public funding, so the outputs of our research should be public. In our case, there is no reason to protect it. So.
And for us, we see that from the other side, it is very interesting how one can produce a very good data for others to like use them. As Monika mentioned, that you have to have the proper metadata, you have to have proper documentation of how the data, the context of how the data were created, and it is kind of tricky to use them straight, like this kind of data, if you don't have this information. But when you do, for example, as I mentioned, the protein data bank, it is very well curated database, basically, with the 40 years of standardization and creating a knowledge around it. So in these cases, it has a very big potential, basically, because many discoveries were created from the structures that are already published. So,
thank you so much. So I have several more questions, but now might be a good time to have a question from Slido.
Just to make sure, there is a QR code now on the slide, so you can always scan it and add a question.
Okay, so we have a first question for Martin. Why 6 months between data provision and data publication? Who decided this period and based on what? Why not 3 months or why not 12 months?
Yeah, very nice question. Thank you very much for that. Firstly, we are speaking about the system of synthetic data and about the period which the requester had for publishing results. And who decided? The management of Institute of Health Information and Statistics. And why? 6 months, because it's enough for getting some really achievements and results on data. And secondly, typical period between the updates is 1 year regarding the particular registry of National Health Information System. So it's enough because after this period, the data will probably be a little bit old. So this is some kind of, let's say, compromise. And finally, we discussed in detail the setup, the rules with the open community of health data, and we agreed on this particular period. But maybe sometimes some words will appear and we will slightly change it. Not sure yet, because we are in the very beginning of the process of synthetic data output releasement.
Do we have some more questions?
Yeah, we have plenty of questions now. So now I would like to join 2 questions into one. One is for Adrián. How is user acceptance of the data management system? Are there any requirements for metadata enrichment? And I believe it might be connected to question which is to Marek. What is the difference between DDR and a traditional shared SharePoint, a G Drive or Dropbox folder? Is it the coupling with metadata?
You were right about the coupling those questions. Data management is practically about coupling it with the metadata, data with the metadata, but also trying to somehow formalize the process of how data are being gathered and then published and shared and this kind of stuff. But you mentioned the first question, it was actually from...
It's the user acceptance of the data management system.
Yes. Well, it is a bit tricky, because if they would have to set it up for themselves, it is impossible, like the research groups or like the users who would like to use it. Therefore, we are building it as an infrastructure service. So it is deployed for the whole CEITEC and potentially for the whole national infrastructure, if we will be lucky and it will get accepted. Then it will be okay, because several people will be managing this infrastructure and they only get the user interface common or comparable to the OneDrive or SharePoint or this kind of services.
For me, yes, it is, of course, this version I mentioned, the light version, which we believe will be on SysNet soon, is not very far from Google Drive. On the other hand, the difference is in metadata, because DDR you can upload JSON and to the predefined data structure, so metadata. And the other advantage in this particular case is also authorization and authentication via eInfra AI. And if especially useful is then to connect it to your internal system, which enables the management of instruments and automated harvesting of data. That's of course the advantage, but that unfortunately requires certain IT knowledge on the side of the user, so that may be a barrier. So that's why we are creating kind of improved Google Drive for everyone, and then with the additional components for those who will decide, okay, we need more.
Thank you. and maybe one more, and then we can continue.
So there is a follow-up question about DDR. So do we want to continue in this topic or not?
Why not?
Okay, so the question is whether the DDR are going to be shared with the community, or is it a closed system for Heyrovsky and Technical University of Liberec? Is it locally hosted?
No, so it's all at the moment it's tested in at Charles University also, and in Department of Mathematics and Physics, and independently at the Science Faculty for one facility, a bioimaging facility. So the system is actually developed to be completely generic, open source. It is on GitLab, and everyone can even contribute to the development or come up with issues which the people would love. But at the same time, as I said, we want to keep it very, very simple. This should be an entrance system to the automated data management.
Thank you so much. So if you don't mind, I can maybe dig in again into the data reuse stories. And we were talking about the motivation factors, and there are, of course, some practical, technical factors, whether you can reuse the data or share the data. And the basic one for me is that the data are well curated. So the question is, how do you persuade maybe young researcher or not so experienced researcher to, let's say, reasonably manage the data?
Let me start with my point of view. What we tried to do is that open data sets as well as data summaries are also the requested part of citation styles. So it means, and we have a deep look on that from our analysis, that it's quite common that scientists, young or senior researchers, are used to citing also the open data sets as a source, which were used for particle or final results. So this is also the way, because you as a creator or somebody who proposed the design of the data sets, you are also you bring also something new, and you must be an author, and you must be cited in an academic community. So this is our motivation.
For me, a lot of the time I feel like I am that researcher who needs to be persuaded to curate their own data. So I will tell you how I try to persuade myself. For me, I mean, in our lab, I feel like we are fairly selfish about this, so I think we represent the community well. We don't really care about these great visions of open science or prestige of having a public data set. We are mostly interested in using other people's data, in exploiting all of these resources and the data that's in the air. And I just think that sharing your data and curating your own data well happens to be a great way to get into that, to understand the systems from the inside. If you just try to just take and exploit the data, it's hard to know where to start, where the databases are, how the annotations work, like what you should even look for. Like a great starting point is to try and share your own data, and that enhances your ability to exploit other people's data. That's like a really selfish motivation for me.
I think it's a great conversation opener. Like when you share your data, like you're building a community, like people will approach you, you will have more collaborations, and if you have more collaborations, you will have more citations. So I feel it's just like, it's not only like good to be like friendly with data, but it also like pays back.
Well, we should distinguish two things: data curation and data annotation with metadata during the early steps, and this is what I want to highlight. It is very important to bring the data annotation to the very early stage of data life cycle. So that's why, for example, in Heyrovsky, but also as a part of Open Science 2 project, we are implementing electronic lab books, which are very helpful in this. This should happen when the data are generated, not when you are publishing it.
Once I got a question at the CEITEC from the research group when I was presenting about the electronic laboratory notebooks. By the way, my background is in the informatics. So I was presenting this, and they asked me, "What's your opinion, or what are you using? What kind of electronic laboratory notebook are you informaticians using actually?" And I was like, "Oh well, we don't need it." And this is the case also within these domains. When I'm creating the software, I usually forgot to write the documentation, and I don't want to write it because why would I? Because I remember what I was writing. So this is also for in the contrast with the publishing data. Many corporates building these lab softwares are also not doing very well with the documentation. So either they should be, or the motivation behind this is either they are credited financially, for example, for the proper support, for very well support, or they are monopole and they don't have to do any documentation because they have the market covered. And I think this applies also for publishing the data sets in the science, that some people want to be credited to create a perfect data set, but the others don't, because they don't need it. They have their own hypotheses. They have their own ideas to create these publications on top of data, and they don't have to publish the data to be credited. And this was something to consider, that this is maybe something that we can help or find out how we should pursue that.
Thank you, all of you. And maybe just to add another question. So, with that all said, to what extent do you open your data? And maybe Jaroslav already told us that they rather use other people's data. But if you want to comment any more, and then we can go back to Martin and.
Yeah, sure. I mean, we're just, as I said, dipping our toes into this area. So we are doing our best. We don't have that many data sets, and we are in the process of sharing them. So we are in terms of data management, we are a very immature group. Well.
Yeah, we open everything, yeah, including the code to generate figures. Yeah.
We are not yet there, but that's. I hope that early next year we are there.
I have again a bit pessimistic story from the CEITEC when we were talking about we have a data, let's open them, let's share them with some colleagues from the Czech Republic or from the Bologna or some else institutes. They asked me, or they were concerned about who is getting actually access to this data, and they want complete auditing trace of who accessed those data, because they can talk to the people and they talk to what kind of publication they want to make out of this and this kind of stuff. And the one of the concern was, I hope that China is not listening to us right now, but the concern was that the China is going to steal our data and we are going to publish nothing, and we will be doomed or something.
Martin, I'll try to specify again. And I was curious because you showed that the data sets which are in the portal, most of them are open, but some of them are not. So maybe if you can comment, like, under which conditions people can enter or download the data.
Okay, sorry, I didn't highlight it. All outputs are totally open. That's it. Only one restriction is you must be, as a new user, registered in a formal way into the system of synthetic data. That's it. Everything else, it's quite open, and we try to be as open as possible. We must be sure that the mixture, if you can imagine, like hundreds of different kind of outputs, like open data sets, data summaries, when you really do some kind of mix, we must be in compliance with GDPR as well as HDS, and then it's a little bit challenging for us because the number of outputs is increasing, definitely, and we must be sure that we will not break this line of, let's say, legislation. That's it.
Thank you so much. So I think we're out of time. Maybe let's just make a quick recapitulation that we all agree that data reuse is needed and we all want it. Some fields are almost there, some are not. And we'll do our best. Thank you so much.
Thank you so much to the panelists, and thank you so much, Jaroslav, Juráček, for chairing this session. Here are your gifts.